Hey guys, I hope you had a good spring break. We're diving into section 2 of unit 8.5, which is genetic engineering. So these are all common biotechnology methods and techniques that most people use. So to do a little background, we have to talk about the difference between bacterial DNA and eukaryotic DNA. Remember, humans have 46 long linear pieces of DNA, but bacteria have circular DNA, which we've talked about before. So remember, the bacteria has a circular chromosome. They also have these teeny tiny little extra pieces that are circular called plasmids. Okay, so we have one large circular chromosome and we have small little pieces of plasmids. Okay, they're both DNA, but this plasmid is kind of like bonus DNA. Okay, so they don't have genes on their plasmids that are necessary for the bacteria to survive. It's literally like bonus DNA. So, like, you can get antibiotic resistance, you can get metal resistance, the ability to actually switch plasmids with other bacteria, which is called conjugation. So that's the ability to reproduce sexually. And the biggest one that's concerned to us is antibiotic resistance. So they can actually get antibiotic resistant genes, they can accumulate them, and then they can basically literally roll these plasmids over to other bacteria. So plasmids are just small circular pieces of DNA that's like bonus DNA. Okay, also in bacteria are enzymes called restriction enzymes. Okay, restriction enzymes are nucleases, which you guys remember nucleases as those little enzymes that chop up DNA, the little DNA ninjas that we were trying to worry about when we were talking about mRNA processing with the 5' gap and the poly A tail. Okay, so a nuclease just cuts DNA at very, very specific locations. So they're like little molecular scissors for us, but they can only recognize a certain sequence of ATGCs, and every single restriction enzyme is unique. Okay, they're named for the enzyme or the bacteria that they were found in. So the most famous restriction enzyme is EcoR1. So EcoR1 is the Escherichia coli or E. coli bacteria. It's the first restriction enzyme they found. Okay, so another one's Hindi3, named for Heliomyphis influenzae. So it's the third one. So literally, restriction enzymes are just bacterial nucleases that cut at specific locations. Their original job was to cut up any foreign DNA from viruses like bacteriophages, but we've learned to use them as molecular scissors to help us. So why do we need to cut open DNA? Okay, we want to cut open DNA so we can make something called recombinant DNA. Recombinant DNA is whenever you can take two different DNA sources and you put them together. Okay, so we're going to make bacteria genetically in the lab to produce human proteins. Okay, so that's the job of recombinant DNA, is taking two different genes and putting them together. And I'll give you more examples in a second. So what happens is you have a plasmid, which is that circular piece of DNA from bacteria. You're going to cut it with the restriction enzyme. You're also going to cut out your gene that you want, your target gene, from human, fish, strawberries, whatever you want. And you're going to cut it with the same restriction enzyme so it fits together, and that's going to make a recombinant plasmid. Okay, and then this plasmid can go back in the bacteria, and then the bacteria will transcribe and translate it just like normal, or translate it, not transcribe it. Okay, and then once again, we want to stick it together, so we're going to use our glue, which is DNA ligase. Okay. So restriction enzymes, when you actually cut the DNA, they're not really cutting. It's an enzyme, so it has the active site. The DNA is in the substrate. So you're going to get two different options. You can get blunt ends, which literally they just cut. So there's no overhang. So they're like, it's just a straight cut, like whenever you cut paper. Okay, what we want is sticky ends. Sticky ends makes an overhang. So this is an example of where restriction enzyme would cut. Therefore, you would kind of have an overhang. So that allows more hydrogen bonding. So when you take your target gene out of the human or the second specimen and you put it into bacteria, the DNA will actually stick together. So we like sticky ends, we don't like blunt ends, because blunt would be like trying to stick two things together like this. We want to stick two pieces together that can hydrogen bond. Okay. Now, how do you make a bacteria that has a human gene in it. So the first thing you have to do is you have to get your target gene. The target gene is the one that you want to translate into a protein. So you're going to cut the human gene with the restriction enzyme, okay? So you have to also cut open the plasmid with the same restriction enzyme, okay? But you got to remember human DNA is linear, so it's going to require 2 cuts to get out the target gene. A plasmid is circular, so it's only going to require 1 cut to open it up. Okay, so we have a human gene that's linear, so we're going to cut it twice. So then we have the target gene. The plasmid is circular, so we're only going to cut that open to open the circuit. Then we can put the linear piece of DNA inside. Okay, we like the restriction enzymes to have the sticky ends, so the hydrogen bonds will literally stick better. Okay, then you add the plasmid back to the bacteria. As the bacteria replicates, guess what else? It's replicating its DNA. So every single time this bacteria replicates, it's copying the human gene as well. So we get lots and lots of copies, and then we can turn the gene on, or it will just naturally translate itself, and we can actually make the human protein. This is huge for artificial insulin for diabetics. Okay, so here's a visual. Okay, we have the human cell. We cut out the gene that we want using a restriction enzyme. So there's our human DNA in purple. We take the plasmid out of the bacteria. We have to cut it again. We only have 1 cut here. Notice there's 2 cuts here because this is linear. So to cut something out linear, you have to cut twice. If it's a circle, you only have to cut once. So then you have sticky ends. So you see this is missing the top half. Well, the plasmid has the top half. So they're literally going to slide into one another. Okay, making the sticky ends hydrogen bond, and your gene will put in there just like a puzzle piece. Okay, so now we have what we call recombinant DNA. Remember, recombinant DNA is whenever you combine 2 different sources of DNA. We're going to put that plasmid back in the bacteria, and then it will start producing. Okay, when you have organisms that have 2 different sources of DNA, we call those transgenic organisms. So they literally have genes across organisms. Okay, so you can put fat cells or the protein to make a lot of fat cells into mice. This is a human ear growing on a rat. Okay, so we literally put the proteins and the genes necessary to grow an ear. So that's for people that get in accidents. The glowfish that you see at PetSmart, those are transgenic organisms. Okay, they have taken a fluorescent gene from other species and they've put it into the fish so you get cool glow in the dark fish. Okay, this is also, they can do it to plants. Some other examples of why we do this is making human insulin. Okay, we can put HGH or the human growth hormone in cows to make milk. So we can actually put a human gene into a cow to make more milk. We can do different genes. We can knock them out to see the effect of different diseases that are crop yields. So they can actually take a salmon gene. Salmon live in really, really super cold water. So they have like an antifreeze protein. So they can actually cut the protein out of the salmon. They put it in strawberries, and strawberries can go into the winter months. So that's an example we use here in the South. You can take corn and you can put a pesticide gene into the corn. So therefore you don't have to spray pesticides. The corn naturally grow. So this is what you guys know as GMOs. And so that's like the whole big debate is you're literally making transgenic organisms, you're recombining DNA, taking 2 different DNA sources. So that's the big controversy with GMOs is, do we know the effect? But literally you're just cutting out DNA and putting it in to a different organism.